Before I get started, Lineage is a publicly traded company, so I'll refer you first to our safe harbor clause, as I will be making some forward-looking statements. So Lineage is a clinical stage cell therapy company. We have 3 programs in early stages of clinical development. All of these are based on our HESC cells. A lot of people say, oh, interesting, an ES cell company, that's good. You don't see so many of those. Well, that may be because we have this very large patent estate that covers that area, north of 800 issued patents and applications. So really just a massive IP estate for this part of cell and gene therapy. What we do is we start with normal human cell lines. This avoids any risks from modifications or messing with the epigenome. These cells are pluripotent. They can become any of the cell types in the body. And what we do is we direct the lineage of these cells to become specific, differentiated cell types. And then we transplant those differentiated cell types into the body to treat whatever ailment or condition it is that we are pursuing. And because of all of our work comes from cell lines, this is essentially infinitely multipliable of these cells. We can generate enough material for even the largest indications. Here's a snapshot of our clinical pipeline. I'd like to orient you to the right side of this slide, which illustrates that each of these clinical programs has received significant support from some validating partners. The Israeli government has put $16 million of support into our macular degeneration program. The California Stem Cell Agency has supported with more than $14 million our spinal cord injury program. And Cancer Research UK has supported more than $10 million of in-kind support and services for our oncology program. I'll begin with cell therapy for dry AMD. Dry AMD, if you've been here today, you have probably seen 1 or 2 presentations with people talking about dry AMD. So I'll just summarize to say, we know that the disease is, the hallmark is the loss of specialized retina cells. So we manufacture specialized retina cells, and then we transplant them to the back of the eye to replace the ones that are lost. Notably, we don't care why those cells are lost. It doesn't matter to us which pathway or what the problem is that is the causal origin for the loss of those cells. We are just manufacturing new, fresh, young, call it RPE cells, and then administering them to replace the ones that are lost. Manufacturing is an increasingly critical component to everything that the companies that are presenting here work on. And I really like this slide. It's illustrative of the kind of control and reproducibility that we have. So this is flow data. The upper 2 panels speak to the identity of the cells, so it's the characterization of the cells, mostly the kinds of surface markers they express. And you can see that we're starting with 0.06% positive cells, so almost nothing in there looks like a retina cell. And then when we're done, 99.5% of them are exhibiting characteristics of RPE cells. In terms of activity, it's very similar. Almost none of these cells are able to phagocytose, but after treatment, we can measure that they're functionally active. So this is critical, having a substantially pure population of RPE cells and being able to do this at pre-commercial scale in multi-liter bioreactors and being able to have very tight control over the characteristics and tight specifications for the release of your clinical trial material. That material is manufactured at our GMP facility located in Israel. We can do parallel processing here. We do all of our development and CTM in this facility. And our clinical trial right now has enrolled 16 of its intended 24 subjects. If you look at the lower part of this slide, the first 12 subjects on this study have the most severe vision loss. They have the most advanced disease. These are individuals who are legally blind. For the most part, the data that we collect from those individuals is anatomical changes. We're looking to see if the cells that we introduce have persisted for a long time. Are we able to identify them? Are they present? And we're looking at the structure of the retina. What does the integrity of the retina look like over time? Can we see some improvements in the structure and the anatomy in the most severe subjects? More recently, we have begun enrolling into the second part of this study. These are individuals who have better baseline vision. They have less advanced disease. The totality of the loss of cells in the back of their eye is not as extreme, and they more accurately represent our intended patient population. The nice thing in particular about part 2 is these are the individuals that have got better baseline vision, so it's a lot easier to look at things like visual acuity and see not just anatomical changes, but also if you're having an effect on the individual's vision. Here's an example of how we're able to monitor the engraftment of the cell. The upper panel, I think this is a FAF image. The upper panel is the baseline, and that yellow dotted line is representing where the cells were administered. It's an area called the bleb, and you can see in the lower panel, 2 years later, all of that darkened region, that pigmented region, that's because these are retinal pigmented epithelial cells, they're RPE cells. So we can just visually look and see that those cells are still present even years after being administered. Here's a case study. We have treated, at this point, only 4 of these better vision patients. No one's going to be surprised this is the best one, but I just want to show you what's possible here. The green line is this individual's treated eye. And in the left panel, we're looking at the visual acuity, so the number of letters that they're able to read on an eye chart. And you can see almost immediately they were able to see, this is about 25 letters more than they could at baseline. That's, call it 4 or 5 lines on an ETDRS eye chart. That is very significant. If you go and Google an ETDRS eye chart and look at the top line and then 5 lines lower or 4 lines lower, you're going to see that that's a pretty clinically significant improvement. And it's been durable. We've reported to date out to 9 months with this subject. And in contrast, the fellow eye had no change, which that was an untreated eye. You wouldn't expect any change. The other thing that we monitored in this individual is the size of the geographic atrophy. So that's literally the region of RPE cells that have died off. You can see them. It's roughly circular, and you can think of this as just sort of like the barren area in an otherwise healthy retina. And it grows. It grows 1 or 2 millimeters, square millimeters per year. And you can see that the untreated eye is growing. This is at 9 months. It was 1.5 square millimeters larger, whereas the growth rate of the treated eye was much slower. So this is what we're trying to accomplish by replacing retina cells into the people who are suffering from dry AMD. We have seen signs of structural improvement. I didn't go through everything, but we've seen signs of reorganization of the retina, elimination of the drusen waste material, and other things that retina cells do for you. And that data is supportive and consistent with our belief that these cells have a benefit. That data has been very encouraging and it's been very well tolerated. We haven't seen any unexpected adverse events from this treatment. We also, just to really hit home a competitive advantage, we also are using a new way to deliver the cells. We're using a flexible cannula that comes around and goes up through a suprachoroidal route and goes underneath the retina to deliver the cells. The standard of care, currently not using this, is to go directly into the eye. You remove the vitreous, that's called a vitrectomy. You puncture the retina, that's called a retinotomy. You administer the cells, remove the needle, and the problem is the material that you administer often kind of floats away. It effluxes back into the vitreous space and can cause all sorts of problems, not least of which is you have no idea how many cells you just administered because a bunch of them floated away. So what we have done is we've partnered with a group, the gyroscope company, to access their Orbit SDS system, which provides a suprachoroidal route, again, coming up underneath and into the subretinal space underneath the retina, does not require a vitrectomy, does not require a retinotomy. It is a stable, precise, and simple way of delivering the cells. And we think in combination with the purity of our cells, the activity of our cells, and now best-in-class delivery of the cells, we think we've got a really nice solution for dry AMD. Why would we want to do this? There are millions of people who are suffering from this, and there are no FDA-approved treatments for dry AMD today. So we encourage you to learn more about the work that we're doing and the exciting clinical data that we're generating, and we even have an update coming up at the AAO conference in just a small number of days. We additionally have work in spinal cord injury. This is Lucas Linder. Lucas was driving his truck. Literally, he was going to pick up donuts for his grandma on a Sunday morning. He swerved to avoid a deer, and he was paralyzed after being ejected from his pickup truck. He was a participant in our clinical trial, and a year later, Lucas was able to throw out a pitch at a baseball game. And he's not the only one of the 25 people that have received our spinal cord treatment to have an improvement, to have benefit. We have people who have gone from not having function in their arms to being able to type as many as 40 words a minute. So just imagine for a moment the difference in the quality of life from being immobile or useless movement to being able to type, right? You can have a life that's very fulfilled and be active on social media and all of the things. You have a job. Like, this is incredibly life-changing benefits that we're trying to confer to these individuals. There's also a massive economic cost to support individuals who require extensive care due to their inability to have a normal motor function. This is a little different. What we are doing in dry AMD is we manufacture retina cells. In this case, for the spinal cord injury, we manufacture oligodendrocyte progenitors. So this is a form of glial cell. These are the cells that are damaged or lost following an injury, and they also provide the insulation to allow those electrical connections to form, which is how you were able to have that motor activity. This program is RMAT and orphan drug designation. The top image is a nice example. This is animal data. You can see that inside that red circle, that giant barren area, all the cells have died off from there. That's an injury that's been placed into a rat spinal cord. After administration of our cells, which are able to migrate and are able to multiply, that cavity is filled in. And we've been able to show in a dose-dependent manner that these animals can recover significant amounts of motor recovery. That's because these cells are secreting positive neurotrophic factors, they're remyelinating the axons, and they are able to stably engraft. Of the 25 subjects, we've had 96% successful engraftment, and a third of these subjects had had 2 or more levels of improvement in motor score. So we're really excited about what we've been able to see in spinal cord injury in the clinic, and it's been an extremely well-tolerated treatment as well. Lastly, thirdly, for us, we're also using cell therapy in a cancer approach. Here, instead of manufacturing RPE cells, instead of manufacturing oligodendrocytes, we're manufacturing dendritic cells. Why do we do that? Because dendritic cells are professional antigen-presenting cells. We can educate them by loading them with antigens and teaching them or telling them to go into the body and attack the tumor. So we've partnered this program with Cancer Research UK. We have 4 subjects treated to date. This concept and where we're headed with this is we think that using a dendritic cell vaccine and being able to take the education of these dendritic cells and in an allogeneic format be able to introduce them and essentially emphasize or drive forward the Th1, Th2 response in patients. We ultimately think that in combination with checkpoint inhibitors, which are responsible for sort of taking the brakes off of the immune system and providing access to the tumor microenvironment, we think those two things together are going to be a beautiful way for this kind of technology to be deployed and drive superior outcomes in cancer. Lastly, I want to mention about Renevia. Renevia is a scaffold. This was originally designed to be a scaffold for cells in thinking about larger deployment of cells and needing to have three-dimensional space. You know, the thing about the retina is these cells lie in a monolayer. But when you start thinking about organoids or larger three-dimensional applications, you really need to have some sort of a scaffold. But the company conducted a study using the scaffold itself in connection with stromal vascular fraction of fat cells, and using that as a filler for individuals that had HIV lipatrophy. The data was very encouraging, and we just a couple of weeks ago received a CE mark for that material. So it's a fourth asset that has been developed from the company. So the company is reasonably well capitalized, either directly through our cash or through equity holdings that we have in other public companies, or through money that is owed to us through transactions that we had done earlier this year. We have been going through a transformation. I will say that this is a company that has an entirely new management team. I am a new CEO for this company. This is my third time being public CEO. I'm a new CEO of this company. We have a new chief medical officer, a new chief financial officer. We have a new general counsel. We have a new vice president of business development. We have a new focus, and we have a new energy. And I am really excited about what lies ahead for Lineage Cell Therapeutics. So thanks very much. And it looks like I've used my time, but I'd be happy to answer any questions you have in the back of the room in just a moment. Thank you.